So I would like to start by thanking Morten and the organizers. It's a great pleasure to talk to the CBM community. It's great that we can do this virtually, and I'm looking forward to the physical meeting next year. So today I'm going to talk about something which I don't usually talk about in these meetings, because I've been talking a lot about the gut microbiota and the glycan metabolism, but this is about oxidative enzymes that are active on carbohydrates and not hydrolases as I usually present. So for those in the community who are a bit younger, who don't know where I am, we are neighbors of the organizing lab. So we are in the north of Copenhagen in the same institute as Peter. And let me see. This is the building we reside in. It keeps the good tradition of technical universities in Northern Europe, but regarding the aesthetics, that is, but we are surrounded by a very nice forest around us. And the sea is not nearly as close as Al Boraston's lab, but it's a bit here to the east. Okay, so I would like to start with my acknowledgements before I forget them, as I have done once in my presentations. So most of the work that I'm going to present today is performed by Majid, who did a tremendously hard work collecting this data for this study. He was also helped by Tina, and this is our last pre-corona meeting, as you can see. This wouldn't be possible, at least it hasn't been possible over the last year or so. Okay, but the data we am presenting is also a collaboration with many other people who made very important contributions to it. So as you know, I'm not an LPMO guy, so this study wouldn't have been possible without a close collaboration with Jean-Guy Berand and his team in Marseille. Bastien Pissarro has been instrumental for designing some of the experiments and for having excellent contribution to the paper writing. I would like also to thank Associate Professor Sebastian from DTU Chemistry for helping us with the NMR, Dit Wellner and Vollmer for the structural biology, Vincent and the Casey team for helping Majid with the sequence analysis and the phylogeny, and Emma Maher and her team in Toronto for characterizing actually one of the enzymes that we are presenting today, and Bruno and Frederic in Marseille for the EPR work we have done, and also the generous contribution from many funding agencies. So I'll start by a brief introduction about LPMOs and about auxiliary activity family 7, which is the center of this talk. And then I'll present our results on this family and how it interplays with LPMOs, and then I'll just make a short conclusion. So lytic polysaccharide monooxygenases have been discovered a little bit more than a decade ago. The first report was by Gustav Weyer-Kolstad and his co-workers in Oos with an LPMO which was active or which is active on chitin, and then this was followed by Quinlan and co-workers with the cellulose active LPMO. So LPMOs are unique because they are able to attack glycosidic bonds in crystalline polysaccharide matrices, thereby circumventing the need to sequester individual change as cellulases that Peter just presented. And this makes them exceptionally useful for opening up crystalline polysaccharides, which are recalcitrant to hydrolytic attack. So they do so by having an active site which reflects the symmetry of the substrate, where you have a catalytic copper cofactor coordinated by a bracelet of two histidines, and this allows the hydroxylation either at the C1 or at the C4 of the bond that is being cleaved. So the importance of these enzymes has led to their classification by Bernard and co-workers as auxiliary activity enzymes in the Casey databases, and there has been a few families that has been discovered in recent years. But importantly, These enzymes are very important in the global carbon turnover and in industrial biotechnology applications targeting recalcitrant biomass. So as I said, I'm not an LPMO person, and actually the way I stumbled into this area was by a very inspiring collaboration with this guy here, looking very cool in this picture. This is Gustav Weyer-Kolstad. And at the time, we were looking at the starch-active LPMOs that Gustav has sort of identified from sequence analysis. We were a bit too slow to report the oxidation activity of these enzymes, but we looked into the secretomes of Aspergillus nidulans during growth on starch. And interestingly, we showed that LPMOs are one of the most highly abundant enzymes in the secretomes. And this work was done by Laura, who visited Gustav and his co-workers, and Morten helped with the proteomics work. And in fact, in this study, we were intrigued by the fact that although starch is a quite simple substrate compared to lignocellulosic biomass, there was a large number of oxidoreductases that are co-secreted with LPMOs and amylases, and many of them are distant orthologs of auxiliary activity 7 or AA7 enzymes in databases. So we were sort of questioning why AA7-like enzymes or proteins are secreted, some of them abundantly in this. And then we're digging into the literature, we realized that there's some more secretomes that contain these AA7s. So some of them growing on different substrates, on lignocellulosic substrates, as in this recent paper. So this was really what triggered our interest in this family. And then looking at this family, the characterized activities there were oligosaccharide oxidases that are able to oxidize the C1, the reducing and the OH groups to the corresponding lactones with the production of hydrogen peroxide. And there were activities on cello, xylo, and chito oligosaccharides reported. So these enzymes are evolutionarily related to the vanillyl alcohol oxidase superfamily of oxidases, which contains diverse enzymes, but mostly with monocovalently linked FAD cofactors. And regarding primary structure, the FAD domains are obviously readily identified. This is a PFAM numbering here in the sequences, but these enzymes have also another primary structure signature, which is a so-called berberine bridge enzyme-like domains. And looking at the three-dimensional structures, the first reported was from this gluco-oligosaccharide oxidase that Emma Master has worked a lot on also. We could identify two structural domains. So this is the FAD buying domain. And this is in blue, the so-called DBE domain, which is not really a structural domain, but it's an alpha helical motif that acts as a lid and packs at the bottom of the FAD buying domain. And this part here is the bottom basically of the active site right under the FAD cofactor. And the second structural domain is the substrate binding domain, where substrates are accommodated here in a deep pocket. So the important thing is that the distinguishing feature of the family, so the hallmarks of the AA7 family, judged by the characterized structures that we looked at the time, was that they are bicovalently, they contain bicovalently linked FAD cofactors, which is not the case in the rest of the VAO superfamily. So looking at the active site topology, as I said, this is rather occluded with the quite deep pocket that accommodates at least the disaccharide, which is consistent with their activity on oligosaccharides and with the fact that they lack activity on cellulose, basically. So we wanted to try to define AA7 because this was a bit vague at the time when we were looking at it. So we had some fairly stringent criteria, and then we regarding sequence coverage, and we had 25% sequence identity to the characterized, biochemically and structurally characterized enzyme. And then we were able to generate this phylogenetic tree with about 1900 sequences or so. Interestingly, the sequences that you see in black here with the coordinate files in bracket parentheses are the previously characterized fungal oligosaccharide oxidases, which all cluster on a single sub-branch in this phylogenetic tree. So there was a lot of space on this tree which wasn't described. And another observation was that Clade I, which contains about 35% of the family, so this is this Clade I, contained previously characterized plant enzymes which have diverse specificities. So these were not only oligosaccharide oxidases. You would see here the berberine bridge enzyme, which is an oxidase that is a part of the synthetic pathway of alkaloids. Here you have the Arabidopsis monolignol oxidase, which is somehow involved in lignin precursor synthesis, probably. And here you would have an oligoglucuronide active enzyme, also from Arabidopsis. So the rest of the clades in the family were basically uncharted, and that led us to do some further analysis of this tree. So what you see here is a taxonomic distribution of the sequences in the family, and interestingly, there seems to be some clustering of the sequences based on taxonomy, or partially based on taxonomy. So Clade I harbors all the plant active enzymes and some other ones. Clade II contains mostly ascomyta sequences. Clade IIb, and that's why we have classified these into two subclades, is populated by Omycota sequences. And interestingly, both these clades are more or less populated exclusively by plant phytopathogens, as many other clades in the family. And then we have the Basidiomycetes clades in clade 6 and 4. So we knew also that the FAD cofactor anchoring is important for the redox profile of these enzymes. So we analyzed that in the sequences, and we were surprised to see that the bicovalent tethering of the FAD did not seem to apply in the family, although it applied for most of the sequences in the previously characterized clade 5A. So, for example, in clade 2A, you would have the cysteine that contributes to the bicovalent anchoring of the FAD completely substituted to other amino acid residues. In clade 2B, the other way around, the cysteine is there in most sequences at least, but the histidine is missing in most of the sequences. So this suggested some modulation of the way the FAD is anchored, and thereby differences in the redox profiles and the oxidative power of these enzymes. So based on this, we selected a few sequences, one from this characterized clade, basically as some kind of a positive control, and then a few others. Many sequences that we selected were not expressing so well, and those sequences which you see with the green circle are those ones which we have produced and carried out some biochemical characterization on. And we determined two structures of these enzymes. So we started out by substrate screening on this library containing saccharide and non-saccharide substrates. The enzymes in this case would have a reductive half reaction, which where the electrons are harvested from the substrate, which is oxidized and then stored in the FAD cofactor. And then the next oxidative half reaction can involve one of two modes. The oxidative mode that involves the regeneration of the FAD cofactor by transfer of the electrons to oxygen, thereby generating hydrogen peroxide, or a dehydrogenase mode, which involves the transfer of these electrons to a non-oxygen molecule, and thereby defining a dehydrogenase mode. And in our case, we have used this quite common redox indicator, DCIP, which changes its absorbance when it's reduced, and we use this for the assay. So this is a sample of the screening that we have made, and what I want to point out too in these graphs is that although these three different enzymes from three different clades or subclades share a common preference to Clade II oligosaccharides, but they display some important differences in the molecular recognition. So, for example, whereas this enzyme from Fusarium is not recognizing the 1,3 bonds in disaccharides which have a glucnac at the reducing end, this is done to a reasonable level by this enzyme. The same with the PB-Ki7A. In this case, we have a more liberal recognition that accommodates an axial C2OH, thereby enabling activity on MAN-NAC and mono-oligosaccharides. So there seems to be quite some functional diversity in the family according to what the enzymes probably have been evolved to in vivo. This is also reiterated from the kinetic analysis, where the apparent Km value, for instance, so these enzymes are classified as oligosaccharide oxidases, and this is due to the very high Km value, for instance, on the monosaccharide glucnac in this case. But with these new enzymes that we have characterized, you can see that the Km on the monosaccharide is a little bit lower than 1 millimolar. So it's more than 20 folds lower than the typical oxidase that has been out there. And this again highlights the functional diversity in the family. So looking at Clade II, we got a typical dehydrogenase, quite reasonable turnover number with the profile on reducing end glucose residues. So this enzyme basically prefers cello-oligosaccharides. And this is again consistent with the kinetic analysis, where we see quite decent catalytic performance on these substrates. However, we were surprised that this enzyme was quite miserable as an oxidase. So whereas we could measure comparable oxidase binding by normalized rates for the other enzymes, in this case, the oxidase activity was utterly low, and it looked a bit odd. And basically, we could estimate this to something of a normalized rate of 10 to the -4 per second or something like that. And this is unprecedented in the family. So this dehydrogenase to oxidase ratio allowed us or merited the classification of this enzyme as the first oligosaccharide dehydrogenase in the family. And these data were also consistent with the NMR analysis, where again, even though we had a 12-hour acquisition time in this case, but there's barely anything happening when O2 is the electron acceptor, whereas when we add DCIP to the reaction, so that the reaction is running in the dehydrogenase mode, there's quite decent turnover taking place. Again, due to these differences, we looked at the flavine signature in this Fusarium cello-oligosaccharide dehydrogenase, and again, it was quite different from the canonical oxidases, consistent with some changes in the vicinity of the FAD cofactor. So we determined the structure of this enzyme, and again, it has the same overall structure and fold as the rest of the family, but it has some important differences. So basically, the active site seems to be more open, which is effectuated by a shortening of a loop at the entry of the active site and a different conformation. And looking at the surface of this enzyme, you can see this hopefully clearly, that the enzyme is more open. And now I'm showing only the other enzyme that we have structurally characterized, but this is valid for all the available structures in the family. Also, the surface is positively charged due to the conservation of several arginine residues in this specific clade. And the second thing we did was to look into the FAD binding domain. Immediately, hopefully, you can see that the cystenyl second covalent link to the FAD cofactor in the canonical oxidases is abolished. We have a glycine in this case. But importantly, we have on the reside, on the backside of the FAD cofactor, important differences also. So this histidine residue is implicated in the activation of O2 that has been proposed to bind at this site here to accept the hydride from the cofactor, and the histidine has been implicated to be important for the activation of this O2 molecule. In the case of the dehydrogenase, this is substituted to a serine residue, and there's also additional changes that make this part of the FAD binding domain a little bit less apolar. And the theory suggests that O2 enters into this site through hydrophobic channels. So probably the whole FAD, or at least this proximity of the FAD cofactor here is not compatible with the same entry of oxygen and activation at the FAD cofactor, which probably have some contribution to the redox profile that we observe. And this is not only this enzyme that we have structurally characterized. When we look at the whole clade, we see that this is a conserved feature. So for instance, this is the cysteine residue that anchors the FAD, and in this case, it's substituted to a number of small other residues. And this is the histidine, which is actually one of the more conserved residues. It's actually the second most conserved residue after the catalytic base in the family. In this clade is substituted to a serine residue. So this is a signature of this clade, which we believe is a clade of oligosaccharide dehydrogenases in AA7. Okay, so we wanted to look, due to the co-secretion with LPMOs and due to the low levels of hydrogen peroxide production, we wanted to look at, to the interplay of this enzyme with LPMOs. This is not, again, my area of expertise, but this is a summary slide from an excellent review published recently on the proposed mechanisms of LPMOs. The peroxygenase mechanism that states that hydrogen peroxide is the preferred co-substrate is one of the factors that encourages us to look into this interplay. But before LPMOs are able to perform their work, the copper 2 at the active site needs to be reduced to copper 1. This is so-called priming activation or priming reduction. And that usually needs exogenous electrons, usually from reductants that are added into the reactions. So we wanted to see if our enzyme is able to sort of work with this LPMO due to the fact that it has the hydrogen peroxide producing activity and the co-secretion pattern. So it turned out that this enzyme indeed is able to increase the activity of LPMO. So even in the absence of any added cello-oligosaccharides, you see an increase in the activity of the LPMO based on the appearance of some double oxidized C1, C4 cello-oligosaccharides in this chromatogram. And when you add increasing amounts of cello-tetraose, which is one of the preferred substrates of our cello-oligosaccharide dehydrogenase, this level of activity observed from the LPMO in this cellulose every cell degradation assay is increased. So we wanted to see if this is a general effect, which is due to the hydrogen peroxide production of the enzyme. So to do so, we looked at the keto-oligosaccharide oxidase from Magnoporthe. And it turns out that this enzyme had low levels of activity on cello-oligosaccharides. And this is something in the range of 2 to the minus or in 3 per second. So we tested the same enzyme in the same assay of cellulose or avicell degradation, and we could not see anything to nearly the same level as the cello-oligosaccharide dehydrogenase from Clade II. So this suggests that this is not only a general oxidase effect, but there's something more that is going on between the LPMO and the AA7 dehydrogenase that we have tested. So there's been several enzymatic redox partners that has been proposed to increase the activity of LPMOs. Cello-biose dehydrogenase is probably the most well-characterized partner to LPMOs to date. And the way this has been described to work is that the dehydrogenase domain of this enzyme harvests electrons from cello-biose or cello-oligosaccharide oxidation and transfers electrons to a cytochrome heme domain, which results in a large-scale conformational change and subsequent transfer of this electron to the LPMO. So, so we wanted to compare the oligosaccharide dehydrogenase that we have, which lacks this cytochrome heme domain, if it was able to do the same thing without any added sort of reductant or exogenous electron source, and then it's also worth to note that cello-biose dehydrogenase also has a low level of oxidase activity that also generates hydrogen peroxide, which contributes probably to the LPMO activity. And this level is probably in the same range as the previous enzyme that I showed in the slide. So the one we tested as a control in our study. So we compared this with the podospora anisarina cello-biose dehydrogenase, and please note that in this experiment, the concentration of the AA7 dehydrogenase is 3-fold lower than the cello-biose dehydrogenase, yet we were able to see a comparable level of LPMO activity on avicell, and yeah, slightly higher native oligosaccharides, but this is only one experiment, and it just told us that the efficiency of the enzyme is at least in the same order of this tested cello-biose dehydrogenase. So looking at the mechanism of this interplay, we wanted to test the effect of hydrogen peroxide on this interplay, and this was done by adding increasing amounts of horseradish peroxidase, which scavenges the hydrogen peroxide, which led to a reduction in the activity. And the conclusion of that is, of course, no surprise that when hydrogen peroxide is not present, the LPMO activity will go down, actually abolished at 80 nanomolar, I believe. We also wanted to test if the presence of superoxide ions that are known to be also produced from FAD oxidoreductases has any effect on the interplay. And again, we saw the same trend with the reduced activity with increased superoxide dismutase concentration. So probably the superoxide that may be produced by the dehydrogenase seems to play a role, at least also in this interplay. So we wanted to investigate if there was any direct electron transfer from the oligosaccharide dehydrogenase to the LPMO. And again, this is with the comparison of the cello-biose dehydrogenase, which had the heme domain that has been proposed to mediate this electron transfer. And when that domain is truncated, the electron transfer efficiency is profoundly reduced in that system. So in this case, we used electron paramagnetic resonance spectroscopy, and this allowed monitoring the redox state of the copper active center in the LPMO. So the blue spectrum here resembles the copper 2 oxidized form. And when the copper 2 is reduced to the copper 1, you have a loss of signal. And in this case, the LPMO was mixed with the tenfold lower concentration of cello-oligosaccharide dehydrogenase and 1 millimolar cello-triose for a short time at room temperature. And then the EPR analysis was carried out, and then you can see a partial reduction of the copper as based on the change on the reduction in the signal and the EPR. Another experiment that has been done was to chemically reduce the cello-oligosaccharide dehydrogenase by substoichiometric amounts of dithionate, and then mix that with the copper 2 LPMO, which is shown here in the blue again. And here again, you could see more clearly that the cello-oligosaccharide dehydrogenase seemed to be able to transfer its electrons and reduce the copper 2 to copper 1 in the LPMO. Of course, this is not complete. This is the complete reduction of the LPMO, again, performed chemically, but it demonstrated that there's some indications of direct electron transfer. So we think that the newly discovered cello-oligosaccharide dehydrogenase is able to transfer electrons directly to the LPMO. We need to look further into that. And we believe that the low level of production of hydrogen peroxide may also serve as co-substrate for LPMOs and may avoid rapid inactivation of the LPMO due to oxidative damage. We believe also that maybe some amounts of superoxide ion are able to also prime the LPMO besides direct electron transfer. Okay, with this, I would like to conclude. So there's a quite large sequence and functional diversity in AA7. There's a quite notable increase in sequences from phytopathogens, and this merits further investigation into a possible role in pathogenesis of this family. We have described the first cello-oligosaccharide dehydrogenase, which seems to have a unique signature compared to canonical oxidases in the family. And this is probably one of the more simple systems to prime and fuel LPMO activities to date. So we are planning to make a more detailed investigation of this interplay, and we are looking more into oligosaccharide specificities, oligosaccharide dehydrogenase new specificities from other parts of the AA7 family. With this, I would like to thank you for your attention.
Thank you, Maher, for a very nice talk. The floor is now open for questions, either by raising hand or type into Q&A. Okay, let me see. Leila, you have a question. So, Simon, if you could open up for Leila Lolegio.
Yeah.
Yep. Come on, Leila.
Hi, first of all, Maher, thank you for the really nice talk. Can you hear me?
I can hear you.
Yeah, I'm on a street walking, so there you go. That's the beauty of
No worries, I can hear you clearly.
Good. So first of all, you are an LPMO person. You can't get away from it. But apart from that, so did I get it right? So is your hypothesis that your AA7 is actually sort of, do you have an hypothesis about the binding site? I didn't quite catch that.
So, I mean, we don't have any supporting data, but we believe that the more open active site of the enzyme may allow direct interaction with the LPMO.
So like it's been suggested for other enzymes.
Yeah, I mean, we don't have any data to support this, but it's notable that this clade, or especially this enzyme, seems to have a more open active site with different electrostatics, and this may allow this may be a site where this interaction, either directly or by transfer route, may happen to LPMOs.
Okay, looking forward then to see the next chapter of you characterizing the interactions. Will be very exciting. Thank you.
Thank you.
Thanks, Leila, and have a happy walk further on. Any other questions? Let me see. Mr. Yang-Su. Hello, nice presentation. One question. I also searched a suitable electron donor for LPMO. Have you compared the difference between dehydrogenase and ascorbic acid?
Yeah, that has been done in all these assays, but maybe it went a bit too quick with the data. So basically, this system is far more, or at least notably, of notably higher efficiency than the ascorbate, at least in the experiments that has been done here.
And Bernard has a question. Do you want to ask it yourself, Bernard?
No, thank you.
Okay. Okay, so
That was not Bernard.
no, Bernard, it's your turn. You have a question. Okay. Do AA7s need to be selective for their oligosaccharide or the LPMO partner, or do they just need to provide a source of electrons for the system?
That's a very interesting question. I mean, this is something that we are intending to investigate. At least what we have shown in this study, that the activation of LPMOs does not seem to be LPMO specific within 3 that we have tested from AA9, which is a good starting point if this is going to be useful. Then it remains to be seen if one can drive this on any type of oligosaccharide substrates, provided that the active site is suitable.
Thank you. Good question, Bernard. And Vincent, you have a question?
Yeah, I have actually 2 if there's time. Hello, Maher, very nice work, of course. I had seen the paper, of course, beautiful. I was just wondering, 2 questions. First, a practical question. Can you exclude that the potential direct electron transfer is due to trace amounts of mediator in the medium, for example, some free-floating FAD? And the second question would be, what do you think is the natural function of this AA7 enzymes?
Yeah. Thank you very much. That's excellent questions. So for this specific enzyme, the FAD is monocovalently linked, and then it has been through 2 rounds of purification. So I don't have any evidence that there's zero FAD, but at least the chances are slim.
Yeah. Okay.
And for the second question. So I guess what you're asking is this function of this enzyme really in vivo to interact with LPMOs? And that I don't have either any evidence for. What has been proposed is that one role that has been proposed for these enzymes in pathogenic fungi is that when they oxidize the oligosaccharides, that neutralizes the elicitor signal that plants have, and thereby they seem to be meant to suppress the plant immune system. Now, if that's the only role, or if they interplay with LPMOs, that's a very interesting thing to look at in phytopathogens, because many of these phytopathogens also have a large repertoire of LPMOs. So it would be interesting to look at this type of co-secretion patterns, co-regulation patterns, and in vitro interplay.
I think it's very difficult to know. There are so many redox enzymes there. It's really difficult to know what are the the co-evolved interactions and what are just coincidences, right? I mean, the work is equally interesting, but it's very difficult. But you seem to agree. Yeah.
Indeed.
Yeah. Thanks, Maher. Thanks.
And
Thank you.
there's a question here from Antoniel Monclaro. If I pronounce correctly. Thank you for the presentation. Are there any dehydrogenases in the AA7 clade belonging to plants, or one that has a BBE domain as well?
So we haven't characterized any, but based on the fact that the conservation of the two residues that tether the FAD, there might be indeed some dehydrogenases because we see some sequences which are possibly monocovalently linked, and even some which are non-covalently linked. So it is possible, but we haven't characterized any.